Hello, this is Ray Enke at James Madison University, and I'm going to give a short video overview of how to set up HISAT2 alignments for eukaryotic RNA sequencing using the CyVerse Discovery Environment. So again, I'm talking about in this video a study that we did in my undergraduate research lab characterizing the mRNA transcriptome of the human retina. We have some central macula and peripheral retina, so some cone and rod photoreceptor enriched samples that we've taken from human donor eyes. These are my three undergraduate students, former undergraduate students, I should say, Scott, Calvin, and Penny, who are all involved in aspects of this project. And here is a figure from our paper describing the RNA-seq experimental workflow. So in a previous video, we had talked about the first step in this RNA-seq analysis pipeline, which is analyzing the quality of the raw sequencing reads. And to do this, we used FastQC, a commonly used software developed by the Abraham Bioinformatics Institute. There's an excellent video tutorial here, or you can watch my previous short video of how to run FastQC in CyVerse Discovery Environment. This short video is going to talk about how to do the next step in an RNA-seq pipeline, which is the read alignment step. That's where you take all of your sequencing reads, and then you align those to a reference genome. In class, we talked about this software Bowtie, which is a type of aligner. It's a very fast aligner. It's very, very good at doing these exact matches. So if you have an exact match, say to a read that entirely is comprised of an exon coding sequence, the software called Bowtie can do that really, really well. What Bowtie struggles with, and this is sort of a eukaryotic RNA-seq problem where you have coding exons as well as non-coding intervening introns. Some of these reads overlap parts of one exon, and then the other part aligns to another exon. And so Bowtie is unable to do this because it doesn't have it's not splice aware, it will just align exact matches. So one of the first pieces of software that was generated to do what's called this splice-aware alignment, factoring in these splice sites, was a software called TopHat. And TopHat can figure out this problem by sort of hashing the read into smaller fragments. And there are a lot of these splice-aware aligners that are available now, like STAR, MapSlice. HISAT2 is the one we're going to be focusing on for this exercise. So HISAT2 was developed in 2016, and again, it is just one of these splice-aware aligners. Here's the citation for the software. And what this video overview is going to do is show you how to set up HISAT2 alignments in CyVerse Discovery Environment. There's an app called HISAT2 Index Align 2.1, and this is a very useful, powerful tool that you can set up using the app. Okay, so now I'm going to switch over and go live to CyVerse. Okay, and here's my CyVerse homepage. I'm going to go first to my data page so I can, as I always do, talk a little bit about file and folder architecture. So I try to have good folder separation here. So here's a folder that contains all of my FastQ files. In the previous exercise, I showed you this folder where I've set up just to have FastQC and multi-QC outputs. I've also set up this folder that has nothing but HISAT alignments in there. So let me click in there and I'll show you that I set up this specific subfolder within the HISAT folder, Demo Human Retina HISAT2, and I'll click in there. You can see there's nothing in there. That's because I just made this folder specifically for this project. So now I'm going to go down here to the apps pane in CyVerse, and I'm going to search up here, typed in HISAT2, and it's this HISAT2 Index Align 2.1 app is the app that we're after. And as always, we sort of get a default name and a default output, and we always want to change these. So I will change this to HISAT2. I'll keep that, but I'll keep the dash, and I'll put human _retina. And then the output folder, again, I don't like my outputs going to the analyses, which is down here, the whole different tab of CyVerse. I'm going to browse for that folder that I created, my HISAT folder. So here's my home directory, my HISAT alignments. And remember, I made this Demo Human Retina HISAT. So this will just, instead of going to the analyses folder, this will go to my data folder in this particular folder that I made. And then I'll select that current folder location. If you want, you can put a comment in here. It's not necessary. I'll just put that this is a demo HISAT2 run. So I've indicated the name. I've indicated the output folder. I'm going to hit next. Okay, so the next step in part 2, analysis parameters, you must input a genome FASTA file. And so what that's asking for is a genome assembly. So in this case, we're aligning to the human genome, and the most current human genome is the HG38 genome assembly. And so that is in the form of a .fasta, F-A-S-T-A file. And what this is it's an enormous sequence file that has all of the bases in the human genome. And so in a separate video, I'll talk about how to find that and import that into your CyVerse account. But to sidestep that, I've actually stashed that file in the community folder in CyVerse. So I'm going to show you how to get to that. So I'm going to hit Browse. And instead of my directory up here, I'm going to change over to the CyVerse community data. These are data folders that are available to everybody in the CyVerse community. If you scroll down to CyVerse training and classrooms and RENKE JHSPH, that's my username, and JMU CGEMS, this is the JMU Center for Genome and Metagenome Studies folder. And in here we have a reference genomes and transcriptomes. And there's one more file, let's see, human. So it's a lot of clicking, but you'll see that clicking is all worthwhile, because now what we have here is the Homo sapiens H38 DNA top level .fa. So that's a FASTA file. And if you can look at the size, you can see that this is an enormous file. It's a pretty much a 60 gig file. And so this is not a file that we want to be moving around. So it's sitting here in the community folder. We're going to select that. And when we select this, this is a critical step for HISAT2, because we have our FASTQ files from some experiment, and we're asking HISAT2 to align those reads. We have to provide the reference genome to align to. And obviously, or it's worth repeating anyway, that if you've generated RNA-seq data from human cells, you want to make sure that you have the human genome. If you tried to align, let's say, FASTQ files from human cells, generated from human cells, to like the Arabidopsis genome, you would have, HISAT would try to do it, but it would be largely unsuccessful. So obviously, you want to match up your sequencing reads to the genome. So we'll select that file. The next thing we're being asked for is to input the FASTQ files that we want to align. And this is a key part where you have two panes. You have a read one pane that's up here, and then down here you have a read two pane. So this becomes a critical part in the experiment where you have to figure out if your FASTQ files are paired end or single end. So if they're single end files, you should only have a read one. If you have a paired end experiment, that means there should be two FASTQ files for every sample, and there should be an R1 file and an R2. And you have to make sure that you put in the R1s here and the R2s here. So something notable, that if you're downloading data from the NCBI sequence read archive, even if those data are from a paired end experiment originally, the sequence read archive often stores them already pre-paired. So if you've downloaded data from SRA, chances are that you'll just have a read one. So you'll essentially be treating it like a single end experiment, even if it's a paired end experiment. So I know that's a little confusing. So we're going to browse for data here. I am going to go back to my home directory. I have my whole folder full of FASTQ files, and I have a whole bunch of Retina FASTQ files here. And I'll just demonstrate with, well, I suppose just these top 4 files. So this top file here is called AMD Cornea 17 S7R1. So that R1 is indicating that this, as I should say, first of all, this is a paired end experiment. And this is the R1 file for this sample. You can see directly underneath this sample, there's a FASTQ file that has the exact same name, except it says R2. So that's an indicator that these two files are from the same sample. They're just a paired end read. One is the R1 read, one is the R2 read. So remember that I'm only inputting R1 reads into this pane. So I'm going to not check the second box. I'm going to go to my next sample, this AMD macular RPE18R1, and let's do one more. Let's do the sample under that. So the file architecture is that if the samples have the same name, the only difference is R1, R2, generally they're going to be right next to each other. So it's fairly easy to just click every other file. Just double check that you're only inputting your R1 files. So for this demo, I'll just select these top 3 R1 files. And what we'll see in the next step is I'm going to go back and select the R2 files for the next pane. But for right now, I'm just selecting those R1 files. And you can see they've all been added up here to the inputs for the R1 reads. Now I'm going to go down here and I'm going to browse for those companion R2 files that are in the same location. And now I'm just going to click on the accompanying R2 files for those 3 files we selected. I'm going to select OK. We can leave the fragment library type. We can just leave it as is. This file type, this is asking you where you have to determine, is it a paired end experiment or a single end experiment? So I've added in R1 and R2 reads for a paired end experiment, so I must select that. This is a required field. Again, I'm going to note here that if you've downloaded files from SRA, even if it was originally a paired end experiment, but you only have one file for sample, that you will need to select single end. So essentially what HISAT is asking you is, are you giving me R1 and R2 reads? If so, select paired end. If you're just giving me R1 reads, then select single end. Okay, all of these other advanced options, I will suggest that you leave them as default unless you have a compelling reason to change them. Hit next. Same thing with these advanced settings, just leave them as default, hit next, and then you get this overview page where you then will launch your analysis. You'll get a notification that indicates when your HISAT job is running, and once it's completed, you'll get another notification, and in your analyses folder, you should get a note that it's completed, and you can go to the output folder. So in my next video, we will talk a little bit about the HISAT outputs and what kind of jobs you're able to set up in HISAT.